All right, so all those different methods need to have some way to verify that they are effective or how effective they are. And there's two widely used methods, the use dilution test and the disk diffusion method. And I'm going to briefly define and explain them both. But this one by far is the most widely used, and this is one we'll actually do in lab in a few weeks. So briefly, the use dilution test is when you put the microbe in question that you want to kill onto something, and then you dip that into whatever you want to use, whatever chemical. So for instance, you'll take some sort of solid support like a metal ring, and you dip it into a liquid culture, and then you let it dry. So now you have the guys you want to kill. Then you put the dried culture into the disinfectant solution for 10 minutes at room temperature. Then you put the rings that have the dry bacteria that are treated into a culture medium and see if they survive. Now, on the other hand, what you can do is sort of it's almost like the opposite. So what you do is instead you take a Petri dish, like the ones we started using last week. And the first thing you do, again, you're going to take the bacteria, whatever it is that you're trying to kill, to determine how effective this particular chemical is going to be. And you put enough of the cells down on the plate that when they grow into colonies, they're going to grow all together in a lawn, right? So how many would that be, do you think? How many cells would you have to put on this plate to generate a lawn? Like thousands, hundreds of thousands. So 100,000 cells were put onto this plate. When the cells go onto the plate, they're invisible, right? So it doesn't actually look like anything's on here. You have to then do what to see the lawn? You have to incubate them for a couple of days so they can grow. And we'll see the effects of growing for 2 days on your plates today. So once you have this lawn, this is what it would look like. Ignore all of these splotchy patches. But if nothing else was on the plate, we would just have this lawn. Now, if instead what you do is you put down your 100,000 cells, then you take a little filter paper soaked in whatever you're trying to test, put that onto the lawn, or put that onto the culture that isn't yet a lawn, and then allow the culture to grow up in the presence of that, then if it's effective at killing that bacteria, then, it's going to kill in some area around the disc. It's going to inhibit or growth or kill the microbe. And it's going to have this sort of halo that is a measure of, it's actually a fairly complicated metric because it's not just a matter of how effective this is. It's also kind of a measure of how well it diffuses through the medium, right? Because if it doesn't get very far, it's not going to have a very high effect. Nonetheless, this is commonly used because most things diffuse fairly well, okay? So we have kind of a gradient of concentration, and at a certain point it stops being effective. And we can measure this, right? The nice thing about it is it's a really easy thing that we can just measure the diameter of this zone of inhibition and get a number that tells us something about how effective this is. So we're going to actually do this. We're going to do this with Staph aureus and E. coli in a couple of weeks, and you'll get to look at how effective different kinds of things are. Some of them you will not have heard of, different kinds of antibiotics. Some of you will or not, may or may not be familiar with. But also we'll look at how effective something like ethanol is, or bleach or something like that, or Lysol. And it's pretty interesting. So another thing to consider is the effective concentration. So if we have the exact same chemical in two different concentrations, and this one is full strength from a bottle, and then we dilute it 1 in 10, and we have this other lower dilution, then what may happen is that this has a really large zone of inhibition, and this is completely ineffective, right? So the concentration is important. What disinfectants do you have in your home? We've already talked about this, right? You've got probably some bleach. Maybe you have some kind of Lysol or some Lysol derivative in a spray bottle. You might have wipes, right? So again, let's just look at where chemicals target microbes. And the three main ones are the plasma membrane, the nucleic acids, and the proteins. So here they are. Here's our very simple prokaryotic cell, which is basically a plasma membrane, this inner blue line. It's DNA. and a bunch of proteins. And here are the different things that will target those areas. So the plasma membrane is targeted by things called biguanides, phenolics, and quats. We're going to talk about those in a second. Protein denaturation can be done by alcohol or aldehydes, halogens, metals, ozone, peroxygens, and phenolics. We will talk about some of those, not all of them. DNA is affected by ethylene oxide, a very effective killing agent, and aldehydes as well. Now, one thing you'll notice is that these three different components of the cell are not all affected. Like if you take something like phenolics, right, it only does two of these things and not the third one. Something like ethylene oxide only does one and not all three. So different chemicals have different subsets of effects, but the three main targets are these things, which are all vitally important for the survival of a microbe. So let's talk about this first class of chemicals. Phenol is the this like ring-shaped structure with a hydroxyl group. This is the chemical that Lister used in his doing field surgery that revolutionized medicine, basically. And what it does is it disrupts the plasma membrane. Now here is a close relative that's been chemically altered in a lab called O-phenolphenol. This is a phenolic, and it was developed because this, while effective, is an irritant. So you most likely are only going to see it now in throat sprays and lozenges, and there are derivatives of phenol that are more commonly used instead because they are less irritating. Another type of derivative called bisphenols are shown here, and two examples that you may have heard of: hexachlorophene and triclosan. Triclosan is a really commonly used additive in liquid soaps these days. They tend to be quite effective at killing bacteria, but because of that, there is some suggestion that they may actually be ultimately harmful for two reasons. One, because are they going to distinguish between normal flora and pathogens? No, they're equal opportunists. And also, you don't really need this level of killing in your house unless you have like an immune-compromised elderly whatever or a child that you're caring for. You don't actually, I mean, we've managed to survive as a species for many centuries without these kinds of chemicals. So this would be something that would be more helpful in the hospital, and it would be nice if it was actually going to be useful in a hospital instead of so widely used that organisms have developed resistance to it. Okay, so here are the phenol derivatives, and what do? They kill vegetative bacteria with mycobacteria. They work if you use high concentrations. They don't really work very well on reliably on viruses. What they do is destroy cytoplasmic membranes and denature proteins. So they're more likely to be effective on what kind of viruses. Remember, there's two main subtypes of viruses structurally are naked and enveloped. So this might be effective against some of those enveloped viruses, but probably not against the other ones. Pros and cons. They have a wide activity range. They're fairly reasonably cost effective, and they maintain their activity in a variety of different solutions. But they leave a residue, which means that they'll have a sort of long-term killing effect, which may not be ideal, especially in your house. Now, some of them are pretty toxic or irritating. Some are non-toxic, like triclosan, which is why it's so commonly used in soaps and lotions. And again, just think about whether or not you think that's an important thing. It's really common now in products, in household products, and in products that will touch your body. So, and it's recent, so we don't really know what the effects are. But I would highly recommend you read the labels and make that decision. All right, alcohol is another commonly used agent. Typically, you'll find it in an aqueous solution, meaning what? What does this indicate? It's been diluted in water a little bit. So it's between 60 and 80% alcohol with like between 40 and 20% water. What does it do? It's very good at killing bacteria and fungi, but not good at killing endospores. It does get some naked viruses. And what does it do? It coagulates, that is, denatures proteins, and it damages membranes. It's commonly used as an antiseptic, right? Because that's when you go get your shot, that's what they use to degerm the area. And it actually is much more effective when it is an aqueous solution with a little bit of water. Pure alcohol is less effective for two reasons. One, it evaporates really quickly. Two, it actually doesn't work as well at doing these two things because pure alcohol is a little bit less polar than the solution. And there's a sort of happy medium. If you dilute the alcohol too much, you're getting close to water, which is harmless. And if you don't have enough dilution, so there's somewhere in the middle, usually around 60 to 70%, that's the most effective. We also have aldehydes, and here are the two most commonly used, glutaraldehyde and formaldehyde. What do you know about this one? What is it used for? Yeah, preserving things. So clearly it's really good at preventing microbes from growing because that's the definition of preserving. So what is it actually doing? It inactivates proteins and DNA by cross-linking functional groups together. Its common uses are in sterilizing medical equipment. So for instance, 2% alkaline glutaraldehyde for about 12 hours is really, really good at killing everything. Sometimes formaldehyde gas or liquid version of it called formalin is used. That kills most microbes very quickly. It's used for bacteria and viruses and can be used in vaccine preparation. And of course, it's used to preserve specimens. Now here's the class that I mentioned earlier called biguanides. Chlorhexidine is the most commonly used. It disrupts plasma membranes. It's pretty non-toxic. It's good at a lot of things, including killing enveloped viruses, but it doesn't touch endospores or mycobacteria. So what disease would it be not particularly good? If you're in a ward for this disease, you maybe wouldn't use this, given that it's not effective against mycobacteria. You're in a TB ward, right? It's commonly found in many skin products and in mouthwash. Now there's also the halogens. This is referring to a part of the periodic table. That is usually referring to iodine and chlorine. So iodine is one of the oldest microbicides. It's very effective. You can either make an alcohol solution, and you've probably seen tinctures of iodine, right? Or nowadays you can get something called an iodophore, where the iodine is attached to a molecule and it slowly releases, because as the molecule decays, the iodine is released. Its main target is the plasma membrane. It's good at killing vegetative cells, but not great at endospores. Chlorine, exactly what it does, despite how commonly used it is, we don't know. What we do know is that there's two main forms. We use it either as a gas, and it turns into an acid in water, and it's used to disinfect a variety of things from swimming pools to drinking water. As I mentioned earlier, it's quite good at killing microbes and viruses, but not protozoan cysts. So things like Giardia and Cryptosporidium would not be touched by this. Also, chlorine is used in this bleach solution, sodium hypochlorite, and 1 to 100 household bleach. It's probably something you've had in your house before. Also, chlorine dioxide is used, and in this form it kills endospores. It's especially good at getting rid of anthrax. And chloramines are this structure here, which is a mix of chlorine and ammonia. What this does is, because the chlorine is covalently bonded to this ammonia, it releases the chlorine slowly over long periods of time. It means that you can use it because the chlorine is more slowly released. It doesn't form as many toxic products as just using it as is usually used in swimming pools. But there are some other toxic byproducts that are not regulated, so it actually turns out to be toxic to fish. And there are quite a few links now to human disease. And I just want to say, again, I want to emphasize that what you can see from that is all of these methods have their strengths and weaknesses, their limitations and drawbacks. Okay, heavy metals. I hope we all already knew that these were dangerous, and I apologize for this typo. But basically, those are things like silver, mercury, and copper. And we are concerned about the bioaccumulation of these in species that eat other things, right? So for instance, if we are consuming tuna or other large fish, then we have to worry about the accumulation of this at higher levels. Because at high concentrations, they're very toxic, so it makes it unuseful medically. But in the past, actually, heavy metals I mean, they're a little bit easier to extract than a lot of these chemicals that we've talked about. And they were used historically and still used in the neonatal unit. Children that are born can actually have, it's really kind of terrible. They can get eye infections from being born because if their mother has gonorrhea as they're exiting the birth canal, it can get into their eyes, and you can use these silver nitrate eye drops to actually treat it. Also, if you've ever had a burn, which I have, you can be treated topically with a silver cream. If you're trying to get rid of allergy, you may use copper sulfate. And basically what all of them do is denature proteins. So here's just an example of a lawn, which has been allowed to grow in the presence of these heavy metals. And if you lift the heavy metal, what you notice is there's no growth under it. Okay, one of the last classes of chemicals we need to talk about are surfactants. And they actually include a very wide range of things. They have all in common this one property, which is they decrease surface tension. Water has a lot of surface tension, so it actually tends to be very cohesive, and we can disrupt that and make it easier to remove things. Soaps and detergents all fall in this category. Soap breaks up like oil into tiny droplets, emulsifying it. Some surfactants, like acid and ionic surfactants, are used in dairy equipment. So here are three sort of main groups of surfactants, and what you can see is they're quite different, and their common uses are different too. So we've got soap, acid, and ionic detergents, anionic meaning negatively charged, and quats, which are cationic, meaning what? Positively charged. Okay. So soap is something you'll have in your house, and it's basically used for degerming, which means what? What is soap doing? What does degerming mean? Removing it, physically removing it. So you're not really killing anything, really, you're just washing it off. So it's emulsifying the oil, it's getting rid of organic material, it's allowing you to wash it off more easily. So it's a great method for just, cleaning your surfaces, cleaning your hands if you're not in a situation where the people around you or you yourself are at high risk of developing infection from some otherwise relatively minor thing. Acid and ionic detergents, those negatively charged, slightly acid detergents, are very commonly used in food service for sanitization. And what does sanitization mean? Remember way back to that list, it's like bringing the levels of pathogens down to some safe level, some like nebulous, mandated by the FDA or whatever organization is governing things. And then quats, these are based on ammonia, and they're positively charged because ammonia, you may or may not know, is a very positively charged molecule. These are extremely effective compared to these other two, and they're bactericidal, so they kill bacteria, they denature proteins, and they disrupt plasma membranes. So we're just going to talk about those for just a little bit longer. So quats, quaternary ammonia compounds. As I said, they're positively charged. Most things that you would use in your house, in your regular life, like soap, detergent, and like the dishwashing detergent, those are negatively charged, anionic. So that's different from these quats. They have the ability to reduce surface tension, as all surfactants do, so they help remove dirt, organic matter, and stuff. Because the quats are positively charged, they do something that your regular household anionic detergents don't, which is they attract the negative cell membrane. Now, why is the cell membrane negative? How can we remember that? What is the charged part of the cell membrane? Phosphate, right? Phosphate on that lipid. The lipid is the hydrophobic part, the phosphate is the hydrophilic part, and what is the charge of a phosphate? It is negative. So if you have a positive detergent, you're going to attract that cell membrane, you're going to be much better at disrupting the membrane. Now, quats are typically non-toxic, so will often be used to disinfect food prep surfaces. They're very good at killing vegetative bacteria and enveloped viruses because of the plasma membrane effect that they have. They don't work on endospores. They don't work on mycobacteria, so you wouldn't use these as your sole method of protecting yourself in a TB ward. And they don't work on naked viruses. Okay, one of the last things we'll talk about is sterilization by gas, and here are the two main gases that are used, ethylene oxide and ozone. Now, let's start with ethylene oxide. It's actually really effective. What it does is it denatures protein. Consequently, it destroys microbes and includes the ability to destroy endospores and viruses, which makes it an effective sterilization technique. So why would you use it instead of autoclaving? Well, there are things you can't autoclave. There are heat or moisture sensitive. It's really good at getting through fabrics because it's a gas, and it can get into equipment and implants that are heat sensitive. Things that are electronic can be treated this way. If you have a pacemaker or an artificial hip, you can clean it. You can sterilize it with ethylene oxide. The limitation is you can't do it on something that will be harmed by it. So if you, for instance, ethylene oxide yourself by accident, you're going to mutate and potentially cause cancer. So you have to be really careful in your use. You don't want to accidentally inhale it. But if you treat some equipment and then you allow the gas to dissipate properly, then it's perfectly safe. Ozone is a form of oxygen that is extremely unstable. It decomposes quickly. So if you're going to use it, you typically generate it right where you're going to use it. You have some kind of ozone generator that makes the ozone, and then the ozone turns back into oxygen. You can use it instead of chlorine for drinking in wastewater. Peroxygens are powerful oxidizer used to sterilants. So peroxygen sounds like what chemical that you've heard of? Hydrogen peroxide. In what metabolic process have we heard about possibly generating hydrogen peroxide on accident? How would you do that in the cell, do you think? Where is water being made in the cell at the end of some process where you're throwing electrons onto oxygen? Electron transport chain. So at the bottom of the electron transport chain in the mitochondria is always the danger that you're going to, instead of making water, create either hydrogen peroxide or superoxide or other reactive oxygen species. And they're damaging. But on the flip side, if you make them on purpose, because you want to kill something, they're quite effective. The good thing about peroxygens is that they biodegrade, so they don't leave a residue behind. And that's important if you're trying to clean something, but then you don't want to just leave something lying around in a film that's going to kill all the other things, even the things you don't want to kill, like your natural body flora. It's less toxic than ethylene oxide, so you don't have to have special ways of containing it. And its effectiveness depends on what you're using it for. Also, it's important to recognize that aerobic cells produce an enzyme that will break hydrogen peroxide down into oxygen and water. So, does anybody here have hydrogen peroxide in their house? What do you use it for? Disinfectant, right? It's a useful disinfectant. It's helpful to know that when it bubbles, what's really happening. When we see that bubbles are forming, when we throw a hydrogen peroxide onto our skin, for instance, and bubbles are formed, what are the bubbles? They're oxygen. They're produced by whatever you're touching, frantically trying to get rid of the hydrogen peroxide by using their enzyme catalyse to break hydrogen peroxide down into oxygen and water. So that will also be something your own cells do, by the way. So for instance, if you have a cut and there are damaged cells, then you pour the hydrogen peroxide on. Some of those oxygen bubbles are coming actually from your own cells. Food preservatives are also an important method of growth control. We can also, instead of adding chemicals, we can use temperature regulation or desiccation, meaning what? Drying it out, right? As we all know, if we take, a steak and leave it on the counter for a day, it's suddenly not so good anymore. If, on the other hand, you stick it in a dehydrator and turn it into jerky, it's good for months at room temperature. So here are some examples. Organic acids are used to inhibit metabolism. That would be things you've probably read if you read the list of processed food ingredients: sorbic acid, benzoic acid, calcium propionate. They all control molds and bacteria, and they're often used in foods, commercial foods, and in cosmetics. Nitrite is really good at preventing endospores from germinating, and so it's frequently added to processed meat. Have you heard of nitrites in meat, like bacon and other kinds of treated meats? It's used because it inhibits Clostridium botulinum endospores from germinating. Antibiotics can also be used to, sometimes they're added to cheese. to prevent those cheeses from spoiling. Obviously, we can use temperature, we can freeze or refrigerate things, but if we do that, what do we have to be aware of? It's not going to kill everything. It's not going to also prevent everything from growing. All right, desiccation, obviously removing water. So we can do this by salting, sugaring, or drying foods. But again, we have to be aware that some organisms are adapted to high salt environments. So if we salt something, we may not get rid of something like, say, Staphylococcus aureus, which is halotolerant. This is a sort of summary of from the most resistant to the least resistant, roughly, of the different microbes that we know of, which ones are going to be the hardest to kill. And you'll notice what's at the top of the list. Prions. Under that are endospores, and then mycobacteria. Okay, so just keep that in mind. Followed by cysts of protozoan like Giardia or Cryptosporidium. Then the actual protozoans, then gram-negatives that are typically more resistant to various things than gram-positives, something that most people find confusing. Fungi, naked viruses, and gram-positive and enveloped viruses. And again, enveloped viruses, most people think, oh, they have this extra envelopes that are going to be more protected. That is untrue. They're going to be more sensitive, actually, because of anything that damages the plasma membranes is going to make it hard for them to fulfill their life cycle, because that lipid envelope is really important for them to be able to get into their host. Are there any questions about this? Okay, how do we kill prions? We have to use an autoclave for an unusually high heat, 132 degrees for an hour, versus how long and what temperature do we usually use for typically for autoclaving things that we're not trying to kill prions? 120 for 15 minutes. Yeah. Or a little longer if we have a big load. This kind of summarizes all of these different compounds that we've talked about and their effectiveness on endospores versus mycobacteria. And you can see that it just varies quite a lot, right? So different things are effective against mycobacteria than endospores. There's also a lot of stuff in your textbook that sort of talks about, summarizes the mechanism of action, which I went through, right? You know, of these different kinds of things, what do they affect? So what are the three main targets of all these chemicals? What are the three things we sort of consider? Remember to draw yourself that picture in your head of what is in a cell, typically, and let's sort of draw a prokaryotic cell. What are the three parts of the cell that we could be affecting? We could be affecting the DNA, or the plasma membrane, or what else? What's a third possible target? Proteins. Okay. And so different chemicals and compounds will have the ability to do one or more of those things. And they'll also have different contexts in which they're preferable to use, and limitations and drawbacks, of course. So here, I mean, this is basically everything we've just talked about. The last thing I sort of want to mention is that it's kind of useful to think about an application for all of this kind of dry information that you very bravely sat through, okay? And let's think about, for instance, an operating room. It has a variety of different surfaces that are going to have more or less contact with patients. It has a variety of different hazards. It has a variety of different things like that need to be treated in different ways. So the way you treat the floor is going to be different than the way you treat the instruments, than the way you treat the bedding, and so on. So contamination is easy. You can see all the different places you could get it, but depending on, you know, where it is and what it is, you would potentially use different methods to take care of it.